It gives me great pleasure to welcome you all here today to our first, what will hopefully be the first of many annual 1KSA symposiums. It's really fantastic to have you all here. A lot of you have traveled from far. We really appreciate that. And all of you are in some way connected to the 1KSA project through being a sample contributor or a sequencing lab. And so it's great to see an expansion of our community today. So we have shared the program already. The DIPLOMICS team is going to kick off with a sort of overview of the 1KSA program, and then we're going to hand over to our partner labs. You're going to give an update of what they've been up to over the last year. And then we've invited 2 PhD students who are using 1KSA data to give an update on their project progress. And then finally, we're going to end up with a guest speaker. So we have Avril Harvey here today from Parsifal and the Southern African Botanical Products Association. I hope I've got that all right. So she's going to be talking after lunch. But in the meanwhile, during tea and lunchtime, please have a chat to Avril about her work. So we're going to kick off with an introduction from Tim. So Tim, for those of you who haven't met him, is Tim Newman is the director of DIPLOMICS and also the CEO of the Center for Proteomic and Genomic Research, which is the host for DIPLOMICS. Over to you, Tim.
All right, well, thanks for being here. So I don't know how many of you were here last year when we launched 1KSA formally at this venue. It's been actually more than a year that we've been working on 1KSA. It was an idea that came up several years ago, and the idea was to initially sequence the big five as more of an attention grabber, but then it evolved into a biodiversity genome sequencing project, influenced in part by the Africa Biogenome Project, but then we realized that was nested within larger global efforts to capture genomic diversity in amongst the global biodiversity. So the Earth Biogenome Project, the Darwin Tree of Life, et cetera. But 1KSA is very distinctly local, and it's meant to capture and not only the diversity of South African species, but the fact that we can do all of it here. And I think this is a very important message that we're conveying to the rest of the world, that we have the competency, the capability, and all the machinery to exploit our own biodiversity. So thank you for being a part of that, because it truly is a community effort to make that happen. So I believe I'm supposed to tell you a little bit about DIPLOMICS. I think most of you already know, but for those who don't, DIPLOMICS is shorthand for Distributed Platform in Omics. We are one of 13 research infrastructures funded by the Department of Science, Technology, and Innovation in a program they have called SARIR. It's the South African Research Infrastructure Roadmap. Lots of titles, very few people know about it. But this is a system modeled off the European Union's program to support science and technology by setting up centers of competence and excellence in various technology platforms. So South Africa borrowed from that now 8 years ago and launched the SARIR program in 2017. And DIPLOMICS was one of 7 entities that was part of the initial rollout of the program, but the only one within biotech, and specifically within biotech, within the omic sciences. So that includes genomics, in our case, proteomics and metabolomics, and then bioinformatics. So we have 2 primary objectives. There's lots of things we do, but you can sandwich them into 2 sections. One is to build a world-class omics infrastructure, and there's a lot that goes into that. And so we have actions or initiatives that we apply towards that, and that includes instruments. So initially we were a very CapEx-heavy entity looking at the inventory of omics instruments in the country. Where do we have gaps? Where can we acquire and place instruments? But then what we realized very quickly is that along with that is understanding whether machines are properly maintained, are they serviced, are they operational. We were very surprised in the beginning that there's actually a lot of equipment. There's a lot of especially sequencing instruments in the country, but most of them were sitting unused, dormant for various reasons. A lot of it had to do with the fact that groups couldn't afford to keep them running. So a big part of our early interventions was to provide maintenance support for those instruments. And then training. Instruments are useless if people don't know how to use them. So training was very important in the beginning and has become almost an essential aspect of what DIPLOMICS is all about. But also it's very important for us to measure impacts. It's not enough to pay for training, pay for service and maintenance plan, or buy and place instruments. We have to show that this is having some kind of an effect or an impact, a return on investment, if you will, from the government's point of view. So all of these are meant to have some kind of downstream impact. So it could be the availability of the latest instrumentation or technology. We want to have reliability in the infrastructure, so we know that it's available and it's operational and it's running as it should. And then, of course, outputting high-quality research. And then the other key aspect of what we do is enable access for students and researchers generally. So accessibility to high tech is actually often a key barrier to advancing science and technology, especially in developing countries. So this includes lots of training. I'll talk about spatial transformation in a moment, and then high visibility, high impact projects, which is why you're here today. So we measure impact in this by looking at skills development, improved access and opportunity. Opportunity is huge. And then reputational gain. It's surprising that actually that is something that the Department of Science and Technology, Science, Innovation and Technology, whatever their name is key for them, because there is sort of this reputation as a developing country or a lower middle class income country that we're just not able to pull these kinds of things off. So having that perception and reinforcing the perception that we've got these skills and capabilities is important. These impacts also come at different levels. Some of them are immediate, some of them take a bit of time to be realized, and others are very indirect or even cumulative, where our impacts are combined with those of other entities to generate ultimately societal, scientific, and economic benefit. And one of the ways that we do that, as you saw at the bottom of the chart, are these high visibility, high impact projects. As an infrastructure program, we've matured and realized you could put the instruments in place, you can train people, you can make sure that everything's running properly, but without a vehicle to make these things happen, that isn't enough. So our high visibility, high impact projects focus on things like method development. 1KSA is a prime example of that, because every genome we sequence is novel. Training, of course. The concept of open science, this is something that the government wants us to promote, so we are pushing that. It's got its challenges, but it's something that's now a core principle of ours. And then building communities, and 1KSA is another example of how we have pooled people together with a common interest and pursuit, which helps us to advance our aims. And that's where I hand over to Shane, who will go into more depth about one of our highest profile, highest visibility projects, the 1KSA.
Thank you, Tim. Okay, so Tim has set the scene and given an explanation of how 1KSA came into being and how the DIPLOMICS team considers this as a high impact, high visibility initiative. And in 2022, DIPLOMICS initially set up a nanopore working group where the labs in the genomics node were capacitated with, at that point, a Mark 1C instrument. And they were instructed to then go ahead and to test different methods on this instrument. And out of that grew, and for other reasons as well, that Tim has mentioned, the 1KSA project, which we then started in 2023. And so there are 6 of the labs that are in the genomics node for DIPLOMICS that are actively involved in 1KSA. They're all listed up here at the top. So we have the DIPLOMICS training lab, we have Promethean 24, Oxford nanopore sequencing machine in that lab, and then we placed 4 P2 solos in the other labs, at UWC, the IMBM, at Sengen, at UPGL, and then also at SciAb. And the SAMRC are also part of the network. And we'll be hearing more details from each of these labs shortly. So many of you have seen this slide before, but I think it's worthwhile going over it again just to set the scene for the project. And just to remind everyone that the 1KSA webpage really encapsulates all the information that we have gathered together for the project. And so the project in essence starts there. And as a newcomer to the 1KSA project, which you guys have already all been through, you would then go to to the web page and to fill in a species nomination form. And this then gets evaluated at the monthly steering committee meetings of the 1KSA project. And if it's approved, and if it's indigenous and there's permits and ethics in place, and if there's sufficient DNA, then it's likely to be approved. The sample nominator is then sent a sample submission form. And I must say at this point that REDCap is the method that we use to keep track of all the information in the project, and the sample submission form is an online form that goes directly to the 1KSA REDCap project. After sample delivery, the samples are QC'd again to make sure, in the case of DNA, to make sure that they meet the criteria required for sequencing. And I'll be talking a little bit more about that in the next slide. And they then undergo library prep. We record all of the different steps that take place in the lab on REDCap. Whole genome sequencing then takes place at one of the 6 labs. And following that, we follow the 1KSA data management policy, where the data is then moved to the CHPC for the draft genome assembly pipeline, and then after that to DIRISA for data curation and storage. And all of these steps, as I said, are captured in REDCap. We record our methods in the 1KSA Zenodo community, so we try to make those as open science as possible, and the bioinformatics pipeline is also recorded in GitHub. A key aspect of the 1KSA project is data sharing and data visibility, and we make the data visible by creating species cards, and we also have videos and stories about the different species that are available via the web page. So in a nutshell, this is the 1KSA project. So where are we at today? This is the landing page of the 1KSA project as of yesterday. And we are very pleased to report that we have hit the 100 sequence genome mark over here in the middle. I'm not sure if this is going to work. Anyway, it doesn't matter. You can see the 100 right in the middle. So these are the key metrics that we record for the 1KSA project. The 5 bullet points on the left-hand side of the slide are all for eukaryotic species, and the one on the right-hand side is for bacteria. And Lonnie will be talking more about the bacterial species later, which we kind of consider a little bit to the one side of the eukaryotic species. But in terms of eukaryotic species, we've now had just over 300 nominations. We've received 120 of those nominations. So there is a big backlog, and we're working, all the different labs are working to bring those samples into the lab so that they can be sequenced. But it's not trivial getting hold of enough sample and making sure that the permits and the ethics and so on are all in place. Then sequencing, as I've mentioned again, is one of the main criteria. We also carry out a draft genome assembly, and this is in essence just to check that the data is good enough for an assembly. And we also then report on the species card some of the metrics from that draft genome assembly. So we're at 78, which is also pretty good, considering that this also comes with its own set of challenges, computational challenges, wall time challenges on the CHPC. But yeah, we're sort of breaking down, well, we're catching up as much as we can to the sequenced genomes. And then the last one right over there on the left-hand side are the number of species cards that have now been published on the 1KSA website. So there were 5 more that went up yesterday for those of you that are looking, so please go ahead and have a look at them. Okay, so what have we implemented, or what has changed, or what is new over the last year? I'm going to talk a little bit about the lab processes now, and then Tina and Trish will talk about the data analysis and the data management processes separately. So in terms of species nomination, we have implemented recently a list of all the nominated species on the web page, which we update on a monthly basis. And this is to enable sample contributors to check that the species that they are interested in, has it been sequenced yet, or has it been nominated yet under 1KSA. The high molecular weight DNA and ethics and permits, those haven't changed. They still are in place. So I'm just mentioning it here because it still remains a requirement. The terms of use for the 1KSA project have also been updated this year. And this is to reflect publication requirements. So as sample contributors want to publish their genomes, we have instigated some policies around how we would like to have this recorded, particularly in the European Nucleotide Archive, as is our suggestion. I'll talk about that a little bit in a few slides' time. And we've also set up a framework for the Data Access Committee. So we've started the Data Access Committee, and the framework for that is now also in place. These documents are also on Zenodo, which you can get to via the webpage if you would like to have a look at them. And then in terms of sample submission, we have also updated the sample submission form recently, and a Word version of the new sample submission form is available on the DIPLOMICS 1K community on Zenodo. Okay, so credit for this slide, method development in the DIPLOMICS training lab, goes to Anya Lagrangi. I don't think she's here, I think she's outside. But she has developed, in essence, this decision-making tree. What we've done for methods is that we have captured all the lab's high-level methods in a single document, which is on Zenodo. And each lab is going to be talking about their tips and tricks that they use in their own lab. So what I'm focused on here, and this is actually what Anya's using in the DIPLOMICS training lab, but she has, in essence, developed a decision-making tree for samples that come in, for DNA that comes in. So the DNA is QC'd up at the top over there. We do make the requirements for the DNA known, but in many cases the submitted DNA doesn't quite meet the requirements. So if it fails, Anya has a suite of tools that she can use for cleaning up the DNA, bead cleanups, cleanup kits, and also a PCR inhibitor kit, which is also proved to be very useful. Then she also will run a tape station, look at the average fragment size, and then based on the size of the fragments, make a call to fragment or not fragment the DNA. And generally, if it's less than 20, the average fragment size is less than 20 kilobases in size, she won't fragment. But if it's larger, then she will fragment, so 40 kb or bigger specifically. She uses an hypodermic needle approach. So she literally just pipettes the DNA up and down 20 times. And that seems to be sufficient for fragmenting the DNA into the right range for library prep and sequencing. She then looks at the predicted genome size. If the genome is less than 1 gigabase in size, the predicted genome size, she'll then look at the amount of DNA. If she has a lot of DNA, and if she has lots of organisms with similar genome size and similar fragment sizes, she will use the native barcoding kit for library prep, which allows you to multiplex multiple samples on the same flow cell. If she has less DNA, so less than 2 micrograms, she will then use the ligation kit, which allows for 1 sample on a flow cell, and she will run it then just on 1 flow cell. She also then looks at the DNA fragment size in terms of the final library prep steps. There's a long fragment buffer and a short fragment buffer. So if the fragments are bigger than 3 kilobases in size, she'll use the long fragment buffer, but otherwise she'll use the short fragment buffer to finish the library prep step. And then dependent also on the predicted genome size, she will then either set up 1 flow cell. So if the genome size is less than 3 gigabases, then if the sequencing goes well, you should be able to get a 20 to 30 times coverage from 1 sample on a flow cell. But if the genome is very big, then multiple flow cells are required. She does use a wash and reload step. So she actually makes 2 libraries, and she stops after about 16 to 20 hours and washes and reloads. And you'll be hearing more about that from other labs as well. At the end, she can then evaluate the output from the sequencing report and then make a decision should more sequencing take place if DNA allows for it. Many of you, she's written to you to say, please can you send more DNA, because now she has to make more libraries. And so it is a bit of a reiterative process. Okay, I'm going to hand over to Tina now. Thanks.
Morning, everybody. So after sequencing, we generate draft genome assemblies, and I'm just going to quickly tell you why that's important. And before getting into the technical aspects of it, it's important to understand why they matter, especially in a biodiversity-rich country, and how they support research, conservation, and sustainable development. So we assemble genomes because sequencing breaks DNA into tiny fragments, and genome assembly is like building a puzzle, reconstructing full DNA sequencing from those fragments. And draft genome assemblies are critical. It's a critical first step towards understanding an organism's genetic blueprint. And draft genome assemblies are a foundation for gene annotation, population genomics, comparative genomics, and conservation. They're not perfect, but it's a powerful tool for unlocking species-level insights. And we focus on Oxford nanopore sequencing technology to sequence all our samples because it produces long reads, which is ideal for assembling complex genomes. And it's better at resolving repeats and structural variants than short read technology, and it enables de novo assembly for non-model species, so no reference genome is needed. Next slide. Thank you. So we use a Nextflow genome assembly pipeline developed specifically for 1KSA, which runs on the Center for High Performance Computing, or the CHPC, and the pipeline automates these key steps required to generate a high-quality draft genome, and it's available on GitHub. So the workflow includes the following stages. After base calling with Dorado, we do a pre-assembly quality control step, which consists of a KMA analysis using KMC, where we'll analyze the frequency of short DNA sequences, which are called KMAs, to estimate genome size, sequence complexity, and coverage. And this helps to assess the quality and characteristics of the data, essential for successful and efficient de novo assembly. To help understand it better, a KMA analysis is like sorting the puzzle pieces before building it. And imagine genome assembly like building the giant jigsaw puzzle without the box cover. And a KMA analysis is a step where you sort the pieces by color, or you sort the edges in one section. You estimate how big the puzzle is and notice if any pieces from different puzzles got mixed in. So the next QC step is to use nanoplot to assess the initial quality of the reads, and we filter out reads using nanofilts, and then we run nanoplot again to compare the quality before and after. And then for assembly, we use Fly because it has demonstrated optimal performance with ONT reads. And to evaluate the assembly, we use BUSCO and QUAST. BUSCO is used to measure completeness using conserved genes, and QUAST is used to assess the overall assembly quality and contiguity. I'll go into that later. So at this stage of the assembly, it still needs to be polished, and we still need to try and correct for any errors that might have come in. We use Recon for this, which also requires mapping the reads back to the draft, using Minimap and SAM tools. And then we re-evaluate the polished assembly with BUSCO and QUAST. Next slide. So as we continue expanding the 1KSA genome assembly pipeline, we're tailoring assembly and polishing strategies to key factors such as genome size, read length, N50, and genome complexity. And recently, we've been working with large plant genomes, which have exposed some limitations of Fly, especially under the CHPC resource constraints. So in response, we've begun testing HiFiism, which is a different assembler, a runtime efficient assembler optimized for complex and larger genomes, especially when read length N50 exceeds 10 kilobases. Initial results have shown strong assembly completeness, and we're currently assessing how best to integrate HiFiism in the 1KSA pipeline. Okay, to guide our approach, can you go to the next? Yes. We follow a read length-based decision strategy recommended by ONT for datasets with reads lengths of, when the N50s are above 10 KB, we continue, or below 10, above 10 KB, sorry. We use HiFiism for assembly followed by one round of polishing. And if the read length N50 is below 10 KB, we continue using Fly, which handles degraded or lower quality input well. followed by polishing. Thanks. Next one. Okay, so our results, the metrics we use to assess sequencing and assembly quality is the number one is the data produced. So this is before assembly, the read N50. So how good are the puzzle pieces that we started off with? The read N50 is the size of the read where half of the total bases are found in reads of that size or longer. And it tells you if your reads are long enough to be useful. Longer reads are like bigger puzzle pieces that help connect more of the image. And then number two is contiguity, and where we look at contig N50 for this. So how connected are the pieces we've assembled? Contig N50 is the length of a contig such that all contigs of that size or longer cover at least 50% of the total assembly. A high contig N50 equals fewer, longer assembled sequences, and that equals to a better continuity. Next slide. And then the third thing we look at is completeness, which we look at BUSCO scores for this. So are all the puzzle pieces there? BUSCO stands for Benchmarking Universal Single Copy Orthologs, and it checks for whether key genes expected in most organisms are present. A high BUSCO score, for example 95%, suggests most of the essential pieces of the genome are accounted for. So far we've successfully assembled almost 80 genomes across a range of species, including insects, fungi, mammals, plants, and fish. And as shown in the figure, our assemblies consistently score above 95%, indicating high quality results. Next. And to showcase the diversity and depth of our work, we've highlighted results from 4 species. So Shane mentioned that we use species cards to, that includes key information like genome size, assembly stats, and conservation status. And we've recently started generating these snail plots to try and also show you all the completeness and the contiguity, everything just in one figure. And I'm going to try and explain it. I tried to explain it yesterday in a workshop that I presented, and it went horribly wrong. So the class participants helped me to, they helped me, they gave me some notes. So I'm going to try and explain it. Okay, so with a snail plot, you can see the BUSCO completeness score in the right top corner, that green pie chart. So you can see the completeness scores for this specific species is 98.8%. And then in the left top corner are the contiguity assembly statistics, all written down. Sorry, I don't have it in front of me, so I can't see, but the first one is the number of contigs, which is in brackets. It would be 200. The scaffold length is the total assembly size in brackets, and then you have the longest scaffold, 3.46 megabases, then the N50 and the N90. So you have it all there, and then those are visually represented in this circle as well. So ignore the colors for now. We'll start at the outer edge of the circle first. So imagine your assembly in a straight line from 0 to 36.7 megabases, like this, and it's just being put in a circle like that. Okay, so that's on the outer edge. You can see that it starts at the top at the 12 o'clock. You go around, and you can see the size increasing around until the total is at the top. Okay, now look at the blues, the 2 blue colors. That represents the GC content of your assembly, which is also indicated at the bottom, the composition. So it's about 50-50, 45-55%. So the sizes are quite equal, the dark blue and the light blue. Okay, and then there's another black circle just below the dark blue, and that indicates the percentage of the assembly size. So it starts at 0, just below the total assembly size, so 0%, and then 10, 20, 30, 40, until you gain 100, the whole thing. Okay, so the colors are layered. So I want you to look at the dark gray parts that are sticking out just in a few places, but just imagine them, they are beneath the orange colors as well. I hope that makes sense. So the longest scaffolds start in the middle, from the center of the circle to the top, and they are arranged from the longest scaffolds, and they go shorter and shorter when you go clockwise around the circle. So they get all shorter and shorter and shorter. And then look at the red one. We'll start with the longest scaffold. So at the point where it says it's almost at 10%, that's where the red line starts, and you go down to the center, it would give you the size of the longest scaffold. So the center of the circle is 3.4 megabases. So this means that almost 10% of the assembly has a scaffold of that length. So that scaffold makes up almost 10% of the whole assembly. Okay, and now we'll look at the dark orange color. So that is the N50, so 50% of the assembly. So it starts here at the bottom, 50%. And then, I'm just going to go a little bit closer. So you would start here, and then to determine the size of it, you would follow where it ends here, and then you follow there, and then that's where you would look at the actual size of it. There isn't a number there, but it's in between 1 and 3.4. And it's in brackets, can't see, but it's 2.0 something megabases. I'm hoping, just stay with me. We're going to go through 3 more, so we'll get there. Okay, and the dark orange is also a layer under the lighter orange. I don't know if that's making you confused. And then the N90, so 90% of the genome, you would look there and then follow the line to see what's the size. You can go around this way or a shortcut there, which is 702 kilobases. All right, can you go to the next one? So this is a snail plot for a different species, for the ant, and you can see it has a little bit more white than the previous one, which means that its scaffolds are shorter than of the previous assembly. So total genome size at the top is 237 megabases. The longest scaffold is a very small part of it, so not even 1% of the genome has a scaffold of that size. Okay? And N50, I can't read it properly, but 473 kilobases, and N90 is just over 10,000, or was it 20 kbs? So first, when you start looking at this thing, always start at the outer edge and then work with the percentages, and then look at what the sizes are of each of the metrics. Let's go to the next one. Okay, so this is for the whale. Total assembly size is 2.36 gigabases. Okay. N50 is at 1 megabases, and the N90 is 202 kilobases. Is that starting to make sense? Is it becoming clearer? We can definitely talk about it later. I have a poster just there if you want to discuss it. And then we have got one more. Okay, so for this one, total genome size, assembly size, 597 megabases. N50 is 205 kilobases. N90, 25.4 kilobases. And remember, it also shows the number of contigs, and the longest scaffold is also, doesn't make up a lot of the assembly. I tried. And then yesterday, we concluded our first genome assembly pipeline workshop, and it went really well. So I just took all the participants through the pipeline that's on GitHub. We did everything step by step, manually, and then I also showed them at the end how to run the entire pipeline using a script. I don't know if they checked the results this morning, but yeah. So it's just the point of it is that when you send us your DNA samples and we do all the data analysis, but it would be nice for you also to know how to do it yourselves. And if you get to sequence more samples, then you can carry on doing it on your own computer using the CHPC. Yeah, thank you.
Thanks so much, Tina. I think we'll be having lots more snail pot conversations.
Well done, Tina. Good job.
Yes, I need that one.
All right. So, we'll give everyone a minute. For those who don't know me, my name is Patricia, and I'm the bioinformatics coordinator at DIPLOMICS, and I sort of manage the data for OneKSA. So, I'm going to fly through this. There are many moving parts regarding the data management for the OneKSA project, and that's sort of depicted in the picture. You can almost see it moving. So we start off with the sequencing devices. They're spread across the network in the dedicated labs. The sequencing data are moved from the sequencing device to an on-site storage system, like a network attached storage, and that's often for short-term storage. And then from there, the data go to the CHPC. Let me take a minute. So this is specifically the raw sequencing data. They get moved to the CHPC, and we use the compute power of the CHPC to run the genome assembly pipeline, as Tina pointed out. Welcome, Anya. And then we also use the compute power to prepare the data for long-term storage. So when the pipeline is complete and the data are ready, the genome assembly output as well as the raw sequencing data are stored on DIRISA. So at the same time, and as we've picked up already throughout the presentation, all these labs and the sequencing and the genome assembly pipeline are generating a lot of data, the metadata. So the sequencing data are not very useful without that. And that's where REDCap comes into play. So REDCap is one of my favorite tools. It's a free software that can be installed if eligibility criteria are met. And it's a very flexible tool that you can design your databases and put systems and processes in place to help with, first of all, to capture and record data, but then also help with admin and operations and project management. So it's very versatile. So REDCap is key to OneKSA. It's a central, in a way, I can say it's a little bit of the backbone of the project. We use it to collect all the species information. The sample submission form is built in REDCap. We use it to track sample delivery and also check the ethics and permits. We capture all the DNA, QC, the extraction, the QC, the library prep, and the sequencing information. All of that goes into REDCap. We capture all the metrics from the genome assembly workshop, so the BUSCO scores, the cost output, and then also information on the storage. So it's all in one central location. All the labs are able to input data into REDCap, so it really allows for a multi-site project. And then, very small, but it has dashboards, so you can visually track progress, and then also very easily extract information. So we extract information for the species cards as well as project reports, and then you can also export your specific variables of interest and put them into your data analysis program of choice. I use RStudio to generate graphs and things for occasions just like this. Then we have learned a lot over the last year, and specifically in the data management side. These are only 4 that came to mind when I was putting these slides together. So number 1, I would say know your IT infrastructure. What are the capabilities and what are the limitations? The ONT sequencing data are very large, and we need to move them fast so that we can make space for more sequencing. You can't afford a backlog in the system. Number 2, data don't manage themselves. So you need to budget for people, for time, for money, and the resources to make it happen. Data management is often overlooked when it comes to large omics data projects. 3, documentation is key. Document how you're doing something, why you're doing something, because we're all human, we forget things, and then there's always the proverbial bus that's around. Then lastly, the systems and processes that you choose to help your project are supposed to enable efficiency. So make them work for you, not the other way around. And that is my little bit.
Thank
Back
you,
to
Tish.
Shane.
Okay, thank you very much.
Oh, you don't need that.
No, yeah, I'll pop it down, yeah. Okay, thank you. So I'm going to wrap up with a few more slides about 1KSA. Just to reflect back on 1KSA being a high impact, high visibility project supported by DIPLOMICS, training is a big component of what we want to provide within the 1KSA program. This is just a highlight of some of the training initiatives we've supported over the last little bit. So when we initially embarked on the PromethION oxonanopore sequencing journey, we had a sequencing workshop, this is back in 2023, in the training lab at DIPLOMICS, and you'll recognize 2 of the participants over there taking part in the training, Nikki and Renata. This led to a further 8 different library prep and training courses since that time. So we trained the trainer through this initiative, and these trainers then went on to reach more than 100 people to date. 2 of those training courses that Nikki and Renata have run were in collaboration with the Africa Biogenome Project, the Southern African workshop, and we'll be doing it again this year when Craig and the SAMRC are hosting the workshop. We've also promoted high molecular weight DNA extraction, and Sengen earlier this year ran 2 very successful plant DNA extraction workshops, and they're planning a 3rd one for later this year. And Anya has also run an insect DNA extraction workshop in the lab at DIPLOMICS. We've promoted open science for the omics community by collaborating with Telarify to generate a sequence of workshops highlighting the importance of open science. And there's a Zenodo link to the slides for that workshop on the slide. If anyone is interested in that and you missed it, please let me know and I can send that to you. And then as Tina mentioned, we had the first 1KSA genome assembly workshop yesterday and the day before that went really well, and we're planning on running that again in September. Okay, so how is 1KSA applying open science? So there are a lot of different criteria that we've tried to build into the project that promote open science. This includes open methods. So we have the website, we have our REDCap records that Trish has described, we have our protocols that are written up in our 1KSA Zenodo community. In terms of open resources, we are enabling access to sequencing. We also have access to the NICA system, both the CHPC and DERISA. We have set up a data access request form on the website. That's something new. And we have a 1KSA data management plan, which is also available via our Zenodo community. And then we are promoting training, as I've just mentioned. In terms of open source, the 1KSA workflow is on GitHub. In terms of open data, I mentioned already the data management plan and the data access committee. We are creating visibility around the genomes that have been sequenced through the species cards. And we are also promoting publication on the European Nucleotide Archive after a period of 3 years. And then in terms of open access, we are also strongly promoting the publication of the genome in open access journals. So in terms of data sharing updates, the terms of use, as I mentioned at the beginning, have been updated recently with information about how we would like the 1KSA genome assemblies and sequencing datasets to be recorded in the European Nucleotide Archive. We have indicated that we would like to see a 1KSA project tag linked to these datasets. And this is really just for us to monitor how many times those datasets are downloaded and used. And that's important criteria that we can then report on to our funders. We've set up the Data Access Committee framework. So the role of this committee is to check publications, that the 1KSA acknowledgement statement is used and that it's correctly, the data is correctly uploaded onto ENA. The other role of this committee is to evaluate third-party data access requests. So on the 1KSA webpage under Get Involved, there is a new form, request access to data, that can be filled in, and that the information in that form will then be considered by the Data Access Committee. So we are starting to think of the 1KSA species in two sort of separate groups. One is sort of a conservation species where release of the data is important for doing comparative analyses. And here we are promoting uploading of the data onto the European Nucleotide Archive after 3 years. We've said 3 years, and we've spoken about this many times before, because we want to give South African students and researchers an opportunity to acquire the skills through doing workshops like Tina's, to analyze the data themselves before it's released into these databases. But we still want to make it as open as possible. And then the other group of species we are kind of calling the bioeconomy species. And so these are species where there could be a lot of intellectual property associated with development of products around those species. And so what we are recommending is that in the case of a bioeconomy species, that the sample contributor, before uploading those data sets into ENA, does extensive stakeholder consultation, both with indigenous knowledge holders and anybody else who might be using that IP, their research office, for example, at their home institution and so on, before making a decision to upload it into the ENA. And we have said that if, under certain circumstances, the 3-year period has passed and there's still maybe an intellectual property process that needs to take place, that they can then request an embargo or a delayed period before that data is uploaded. And so we're trying to balance open science with access where necessary. I mentioned the data transfer agreement. We also have set that up as a template, and this is for when third-party scientists require that or receive the data. So it's an agreement between those scientists and 1KSA. And I just wanted to mention that we are very much aware of what's happening internationally. So the COP16 meeting occurred late 2024 in Colombia. And one of the outcomes there, this is the UN Convention on Biological Diversity meeting that happens every 2 years, is that new rules were adopted which decouple access from benefit sharing. And specifically, this meeting has set in motion the establishment of the so-called Cali Fund, which is going to be a requirement for commercial users of biodiversity data to contribute to that fund going forward. So this is an evolving space, and it's something that is obviously applicable to 1KSA, and we're going to be watching that going forward. Okay, so what impact has 1KSA made over the last year? So we are a proudly South African project. We are determined to do everything in country, to keep it in country: sampling, DNA extraction, sequencing, draft genome assembly, and also data storage. We've already mentioned in the first year, we've sequenced 100 eukaryotic genomes. This has generated more than 6,000 gigabases of data, 78 draft genome assemblies, and 534 bacterial genomes as well. And this has been done by Lonnie at UWC, and Lonnie will be talking about how they are a biodiversity biobank South Africa partner. I hope I've got that right, yeah. Importantly, 1KSA is an example of a distributed project. There are 6 labs involved. We have also developed a community of practice. This is an evolving space. We update our protocols as we go along, and we share those on Zenodo and GitHub. We have generated a lot of experience with the nanopore sequencing. So not only does this enable leverage of these methods for human genome sequencing projects, but it's also enabled our partner labs to be able to get involved in other projects outside of 1KSA and to offer services as well. Importantly, we're a community project, and 45 South African scientists have nominated eukaryotic species to date. So that's 45 individual scientists that stand to benefit from a draft genome assembly already in the first year. And just to highlight that impact, these are all the organizations where we have had sample submission from, and this is in the first year. So we are growing, and we're very happy with how people are taking up this project for their research. So what's next for 1KSA? So we've recently signed a memorandum of agreements with SANBI, and you'll hear a little bit more about that later, and also the Two Oceans Aquarium Foundation. We have representatives here from TOEF, so we look forward to working with them more going forward. We are wanting to develop sample submission pipelines, so we want to increase the speed at which samples are coming through. I spoke about the backlog earlier on, but we want to engage with different organizations to bring groups of samples through at a time. We want to expand our working groups, for example, a plant working group, a bird working group, fungi, mammals, and so on. So this is to evaluate the biodiversity of South Africa and to see where there are gaps in 1KSA and to try and close those gaps by identifying species that can be sequenced. We want to work closely with the biobanks and the National Collection Facility. Long term, we would like to integrate the genome sequencing data with other biodiversity records, which is why it's so important for us to collect all the metadata around the sample that is sequenced. So there is GBIF, the Global Biodiversity Informatics Facility. SANBI is part of GBIF, and they've indicated that in 2 years' time they would like to bring genome sequencing on board. So we're starting to have conversations around that and how 1KSA data can be included. We want to continue being part of the evolving access and benefit sharing discussions. This is an evolving space, and it's very important that 1KSA is aligned to what is happening internationally. And then we have stated right from the beginning that one of the outputs that we would like to do is to develop a biodiversity genomics education program, and so that still very much is on our to-do list. I would like to say a huge thank you to all the 1KSA partner labs. This is really a partnership of many, many people, and we would not be able to proceed with all the sequencing, all the work that everyone has done. I also would like to acknowledge the DSTI and Syria's funding, which has made this possible, and to thank Tim also for making this possibility available. This project really is, just to go back to what I said at the beginning, a proudly South African project that is benefiting lots of different people.